Welcome! I'm Kerstin from our NIPPON Genetics Lab in Düren, Germany. In today's webinar, we are going to demonstrate the qFYR Analyzer Studio. So let's start with software installation. So the software installation file can be found on the included USB flash drive. Open the file by double-clicking and execute the software installation. Confirm by Next. And then choose a folder and location where to install the software. Confirm by Next. To install, just click Install to start the installation. Wait a few seconds. In the next step, you have to pre-install the drivers for the software. Therefore, click Install and install the selected drive. Once the process is successful, confirm with OK and close the window. Click Next and finish the software installation. To open the software, double-click on the software icon and a login screen will appear. Here you can either log in as guest or as any other predefined user. To predefine new users or update these, just log in as admin. The preset password is admin. Please change this as soon as possible. Just click Login. And in the next window, just select Users to update or define new users. Here you can either add, update or delete users or change passwords. Confirm by OK. If you just want to start an experiment, you can log in as guest. Just click Login and the experiment setup screen will open. Here you have two options to set up new experiments. You can either click Experiment Setup in the lower right corner or use the toolbar and select New Experiment, Experiment Setup. Next to setting up new experiments, you can also open predefined experiments. Just click Open or use the toolbar and select Open. Now we're going to set up a new experiment. Thereby, just click Experiment Setup. Now the experiment properties window will show up. Here, the experimental properties are defined. First of all, you can define the name, which is by default today's date and the time of the start of the experiment. Next, you can select the plastic type. The qFYR supports transparent or white plates or low-profile tubes. In the next part, you can define the experiment type. This can be selected according to the desired analysis. So first of all, a standard curve is an absolute quantification based on a standard curve is used to determine the actual concentration or copy number of target genes, as well as the amplification efficiency. Here, a template solution of known concentration is serially diluted several times and a standard curve is generated. The CQ value of an unknown sample is then compared with the standard curve to determine its quantity. Next to a normal standard curve, you can also use a relative standard curve as experiment type. This method uses a set of relative standards from which unknown samples are quantitated. Here, quantity is expressed relative to some basis sample, the reference sample and target. This sample or target is used as a base for comparing results. For example, in a study of drug effects on gene expression, an untreated control would be an appropriate reference sample. For all experimental samples, target quantity is determined by interpolating from a standard curve and then dividing by the target quantity of the reference. The reference then becomes the 1x sample and all other quantities are expressed as an n-fold difference relative to the reference. Next to standard curves, you can also do a comparative CQ experiment or the delta-delta CQ method as experiment type. This method is a very common method of relative quantification analysis. It compares the results of experimental samples, including controls, such as untreated or wild-type samples, and normalization targets, such as housekeeping gene expression. Using this method, the CQ of the target gene in the same two samples can be compared based on the CQ of the normalization gene in the test sample and the control sample. The resulting delta-delta CQ value can be used to determine the fold difference in expression. The next experiment type that can be chosen is a melt curve. If you choose this experiment type, the melt mode can be further determined. Here you can either choose nucleic acid or thermostability assay. The nucleic acid melt mode is used to determine the melting temperature of the PCR product. Gene sequences with different base compositions or length have different melt temperatures, so different reaction products can be determined according to this value, including non-specific product. Next, you can choose the thermostability assay. This assay is used to analyze protein thermostability and to identify optimal conditions or conformation sequences that favor protein stability, including the investigation of protein-ligand interactions. Analysis based on temperature-induced protein denaturation and the melt temperature is taken as a reference for the thermostability of protein of interest in different conditions. The next experiment type that can be chosen is a high-resolution melt. During a high-resolution melt analysis, the change in fluorescence is detected at very small temperature increase steps, allowing the identification of SNPs and mutations. For detection of unknown or new mutations, saturated DNA dyes are usually used to genotype by high-resolution melt curves. The next type of genotyping experiment you can choose is just normal genotyping. For the detection of known mutations, the most commonly used basic method is the endpoint genotyping using enzyme cleavage hydrolysis probe. Here, 4 primers and 2 probes specific for each allele are used in the multiplex mutant and wild-type allele assay. In this assay, fluorescence is analyzed at the end, and the fluorescence intensity of wild-type specific fluorescence tag in each sample is plotted against that of the mutant tag. Homozygous mutants and homozygous wild types are clustered along their respective axes, whereas heterozygous clusters along the diagonal. The last experiment type you can choose is a presence-absent experiment. This protocol is using a probe-based assay to control the presence or absence of a target, and samples are analyzed by the quantity of target collected at the end of the PCR reaction. As a last step, the used chemistry can be chosen. You can either use probe-based or dye-based experiments. After defining the experiment properties, just click Next to enter the method section. Here, the pre-designed method for each experiment type is shown. Every step can be adjusted according to individual protocols. The default volume for the reaction is 20 microliter. Volumes starting from 1 up to 50 microliter are supported. Temperatures of each step can be adjusted by either dragging or double-click and write the desired temperature. Just put 55. Times of each step can be adjusted by writing. Just double-click and put in the desired time, or use the arrows to lower or increase the time. A green highlighted and activated camera icon indicates the fluorescent is required here. Just click on the icon to activate or deactivate. The ramp rate of each step can be changed by using the drop-down menu. You can choose ramp rates in 0.5 degree depths. If you would like to add or remove a step, mouse over the desired position and click plus or minus. Minus to remove the step and plus to add a step. You can either add a step before the selected step or after. If you would like to add or remove a whole stage, mouse over the desired stage and click plus or minus. Minus to remove the whole stage, or plus to add a new stage. Melt curves can be acquired with different temperature intervals, which can be selected here by drop-down. By clicking Next, the software enters the plate setup section. The plate setup interface is used to set the properties of the reaction wells, including assigning samples, test targets, and reaction well tasks, as well as other properties to each reaction well. This will make the identification of the sample and the subsequent analysis possible. So here you have the sections for either the sample setup or the target setup. To add a sample or target, just click Add in the according section, and the next sample or target will be added. To remove a sample or target, just click X next to the target or the sample. Action has the option to save samples or targets to a library or import previously saved items. Just click Import from Library and import previously saved samples or targets. If you want to add a target or a sample to the library, select the target, click Action and choose Save to Library. For relative quantification experiments, the reference sample and reference target can be assigned using drop-down menus. To assign samples and targets to the plate layout, just select individual wells and click on the sample and target that should be assigned here. For each well, the task can be chosen in the target setup, down here. The default is undetermined for unknown samples. This can be changed according to the desired task. In experiments using standard curves, standards can be set up by either clicking Action in the top right corner or by right-clicking in the plate layout and choose Define and Set up Standards. In the pop-up window, the standard can be defined and arranged in the plate layout. You can define the number of points, the desired number of replicates, the starting quantity, as well as the serial factor of the dilution for the standard. The software will either select and arrange the wells for the standard automatically, or you can choose Let me select wells and select the wells where you want to add your standard. Click Apply to transfer the defined standard and then close the window. Wells are now marked with an S, and after selection of one well, in the target section, the assigned quantity will be displayed, as you can see here. By clicking Next, the software enters the Run section. First, insert your plate to the instrument. Remove the dummy plate and insert your plate and close again. In the Run tab of the desktop software, click Start Run. After clicking Start Run, a Save dialog box appears. Select the data file storage location, name your experiment, and click Save. The run will be started, and the real-time heating temperature and reaction plate temperature will be displayed in the upper left corner of the running interface. Once the heating lid temperature of 100 degrees reached, the estimated runtime will be displayed in the upper right corner, and the cycle number of the reaction will be displayed in the upper left corner. During the run, the real-time acquired fluorescence will be displayed as multi-component plot in the Run section. Once the run is finished, a pop-up window will be shown. Confirm by clicking OK, and the software will enter the results section. FastGene qFYR Analysis Studio will analyze experiment results by default settings after an experiment is finished, and show the amplification plot in the Result tab, as well as the CQ results in the plate layout. In the plate view, the information of each well, including target, task, dye, and sample, will be showed while the mouse is moved over and paused on one well, like this. To display the results as list, click here. All the experiment information is listed, including the well, sample name, target name, task, according dye, CQ, the melt temperature, and so on. For omitting wells, right-click on the according well and choose Omit. After omitting wells or changing any settings, please click Analyze to obtain the new results. Like this. Overview the amplification curve and evaluate the curve shape. A typical amplification curve includes 4 different parts. Down here, the baseline stage. Here, the exponential stage, a linear stage, and a plateau stage. The plot can be adjusted by using the toolbar right above the graph. Here, it can be selected whether all or individual targets shall be displayed, or what plot and graph type is shown. You can change between linear and log graph types, and the plot type, and you can choose which plot color shall be shown. You can either select the target color, the sample color, the well color, or the flag status. If one individual target is selected, the applied threshold will be shown. By opening the plot properties, it can be selected if an automatic threshold should be shown by clicking here, or if the threshold can be adjusted by the user. Each graph can be separately printed or copying by clicking here for saving the graph, here for copy the graph, or here for printing. Graph properties like text, font and text color, as well as the axis labeling and range can be adjusted here. Next to the normalized amplification plot, the raw amplification plot is given. Here, real-time fluorescence signal change during the PCR stage is shown. In order to view the real-time fluorescence signal of the melt curve stage, enter the raw melt plot. The according melt peak plot can be viewed by entering the melt peak plot tab. Per default, the derivative reporter plot is shown. This plot displays the first derivative of fluorescence signal changed according to the temperature. The melt temperature, or the temperature where 50% of the double-stranded DNA are unbinded to single-stranded DNA, could be used to detect unspecific amplification in primer dimers. For example, like here in the negative control samples, you can see different curves than in the rest of the plate. So this seems to be non-specific product or primer dimers. In addition, the normalized reporter can be plotted. Click here. This displays the fluorescence signal value decreasing as temperature rises. The tab QC Summary gives an overview of all applied flags, with the accordingly flagged wells. To see which wells are flagged, just click on the flag, and the according wells will be highlighted. In this webinar, an overview of the most commonly used experiment types, the standard curve, and a comparative CQ experiment will be given. For the experiment type comparative CQ, next to the previously discussed amplification plots, a gene expression graph will be displayed, together with tabular view of the results. Therefore, just enter the gene expression tab. The software will automatically determine the delta CQ delta delta CQ, as well as the RQ, which equals the 2 minus delta delta CQ value, according to the previously defined references. The experiment results are given either as graphs, or you can view all the values of each result just here in this table. There are two plot types available, the RQ versus sample or RQ versus target. In the RQ versus target plot, the relative quantification values are grouped by target, and in the RQ versus sample plot, the relative quantification values are grouped by the samples. General analysis settings can be changed by entering the analysis settings menu in the toolbar. Choose Analysis and then Analysis Settings. Enter the tab Relative Quantification Settings to edit settings used for relative quantification of data, such as references, efficiency of use primers, outlier rejection, and the RQ min-max calculations. Efficiency of the used primers is typically calculated in a standard curve experiment. So let's open a standard curve experiment. After defining a standard in the plate setup, the qFYR Analyzer Studio will automatically generate a standard curve that can be viewed by entering the according tab called Standard Curve. To view all the stats, just select an individual target by clicking here, and the amplification efficiency in %, slope, y-intercept, r-square, and error will be shown below the graph. A slope of -3.2 to -3.5, an r-square of higher than 0.99, and an efficiency of 90 to 110% are indicators of a reliable experiment. The default mode shows red dots as standard samples, while blue are unknown. And you can change the plotted color by selecting a sample, target, or task. After reviewing the results section, click Next in the lower right corner to enter the export section. Here, data can be exported to XLS, CSV, or text files. The default file name is the experiment name that was defined in the Properties section. But you can change the name according to your needs. Next, you can choose what content should be exported by either just checking the predefined boxes or even customize the exported content. In the Customized pop-up, you can select all the content that should be exported by individual columns in the Excel. Click Export in the upper right corner and the exported Excel file will be opened. But first, let's choose a location where to save our export. You can either use the predefined location or browse other locations. Then click Export. And the exported Excel file will be opened. To review the exported Excel file, just click through all the tabs and it will contain results, the run information, sample setup, amplification data, as well, if selected, the melt curve data. Next to the export as file, a report containing graphs can be generated. Click at File in the menu toolbar and select Print Report. In order to report your results, make sure that all wells are selected prior to clicking Print Report. Choose the results that should be exported and either click on Print Preview to preview or save the generated file, or Print Report to directly send the report to a printer. By selecting Print Preview, the report will be opened and can be either saved as encrypted file or sent to a printer. Just click Save to save to the desired location, or Print to send to a printer. And that's it. That was today's webinar on the qFYR Analyzer Studio. Bye.